The following audio recording has been designed for learning purposes only. Do not share, distribute, or provide any type of copies, or this audio lesson will not be made available to you. Good luck with today's lesson.
What I want to do first is talk about what's happening this week into next week. Today in lecture, we're going to finish chapter 34. I think this is where-ish we left off. I'll get some, okay, it looks like that sounds good. We're going to finish chapter 34, roll right into chapter 10, which I don't think we'll finish today. That's why we've got Wednesday to finish it off and do a little tiny part of chapter 42 as well. That is getting us ready for a week from today. Next Monday is exam 3. Okay, so you know I always try to talk about stuff before it happens so you don't get caught off guard. So a week from today is exam 3, which covers chapters 9 on mitosis or the cell cycle, the little teeny tiny part of 31 that we did about homeostasis really and like ramping up from tissues to organs, chapter 34 on the digestive system, and then chapter 10 that we're still going to do, which covers meiosis, and chapter 42 that talks about what happens once you've got the sperm cell and the egg cell that have successfully combined. Where do you go from there? So that's what's coming up in lecture. In terms of lab, I think I reminded you guys last week, today we are starting our rat dissection. So we have 3 days in lab to do this one lab, which I always stress. I'll stress it when we get over there again, just to make the point about how important it is. We've done an entire lab before in 1 day, and this time we're taking 3 days to spend on one lab, so you know it's going to be a big, involved, important one that's going to show up in a big way on like the final lab practical, which seems crazy to talk about because you literally just took the first one, but you should already mentally shift gears. What do I have to do to prepare for the final one? Because the rat lab is the first lab that will be on the final lab practical. So I'll talk to you about that when we get over there. I want to ask, maybe really quickly, for anyone that might have missed it last week, does anyone need a copy of the Chapter 9 assignment? Remember, you guys are supposed to work on it as homework this weekend. It'll be due today at the end of class, but I want to see if anyone needs a copy. Okay, cool. We can take a couple minutes and talk about it later if you guys want, after break or whatever. The other thing I want to pass out is a new handout about the writing assignments that you're going to do for the course. I posted a little sneak peek. I don't even know if people saw it, but I posted a little bit about this on Canvas, maybe Friday or Saturday. I can't remember when it was. I'm not going to go over this right now because I know we're missing a lot of people. So I'm just going to pass them out to you guys. You can look them over. And then on Wednesday, perhaps, we'll take some time and talk about it to make sure you guys know like what is going on with these. Your first one is due March 31st. So you've got several weeks. This is not like a 20-page research essay kind of deal. It's supposed to be, I don't want to say short and sweet, but relatively short and sweet. The gist of it is you're just going to find some kind of article. You could just be like on CNN or like NPR or wherever, some kind of article that relates in some way, shape, or form to biology. Any topic in biology. So pick something you're interested in, and you're basically just going to summarize the article and talk about how it relates to class. Okay, that is it. So, like I said, we will go over the details about it probably on Wednesday. I just wanted to get you the info so you could look it over if you want and have questions prepared for Wednesday if you're confused about what's required for it. Any questions on that quick little one-sentence overview of what this assignment will be? Okay. So you know what's going on in lecture. Talked a little bit about lab, reminding you that we're doing the rat dissection. And I gave you the handout, so I think we're actually ready to get the ball rolling with what we're talking about today. I think we talked totally about the stomach, and we were just about to transition, or maybe just had transitioned, into thinking about the small intestine. Before we just dive right in, you guys know I like to try to do a little bit of an overview to bring us back into the fold. So maybe I'll talk about all of Unit 3 that we've done so far. Remember the main idea with Chapter 9 was we learned how to make more cells, right? We have already spent a lot of time talking about cells, like the parts they have, how they're doing all the work they need to do, all of that. And so we're ready to kind of move up in that big chart from cells to organs, cells to tissues to organ systems to really understand an entire organism. And In chapter 9, we took the step we had to take before we could jump up to looking at tissues, which is how do you go from one cell, which is really all we've thought about in here before, to making more cells like that, enough that you can actually build the tissue out of in a multicellular organism like ourselves. Again, chapter 31, I cut back substantially from what was in the notes that were posted. If you have any questions about that, make sure you come see me and I'll tell you what we crossed off if you missed out on that. The little bit of that chapter we did was just acknowledging that you combine different cell or the same kind of cell to make tissues. You would then combine multiple tissues to make an organ. And every organ, although it's doing an important job, can't do everything on its own. That's why our organs are put together into organ systems that are all working together to accomplish some major body process like digestion or like respiration, like circulation. So we looked pretty quickly at that big overview table, a list of the different organ systems and what process they're involved with. And then the only other thing we really talked about in chapter 31 was homeostasis, which we had heard a bit about in chapter 1. Remember, homeostasis is how we are constantly monitoring our internal conditions for any any condition you can imagine, and making adjustments as needed to make sure we can stay alive. So we looked at the fact that most of that regulation happens with negative feedback regulation, right? The whole idea that if you get too cold, your body works to heat you back up. If you get too warm, your body works to cool you back down. We're always trying to get back to that set point or like sweet spot where we're supposed to be. That was our, bless you, that was our way of transitioning, thinking about tissues and organ systems. That was a lead into chapter 34, which is what we're doing right now, of course. We're looking at our digestive system, just as one example of how this whole idea of organs and organ systems really works. Again, I always include this one because it's a chapter or a body system that most people know at least a little bit about. So some of this, I hope, is sounding a little bit familiar. I also always include it because I hope you'll find it will help us once we get our rats really kind of opened up on Wednesday and start looking for where these organs are. So what we've been doing so far in chapter 34 is essentially following the path of food. We're just tracking where the food is going and what's happening to it as it's moving through the major organs of digestion. Remember, it's considered a major organ of digestion if the food passes through it at some point. So far we've seen that we start chewing our food in the mouth, of course, then it mixes with saliva. And I'm mentioning that specifically because remember, we learned that digestion really happens in two different ways. We've got mechanical digestion, where food molecules or food chunks really are broken down physically into smaller pieces. But we also have to do chemical digestion, which requires the use of specific enzymes to break down big molecules into smaller pieces. And in the mouth, our first organ we looked at, both types of digestion begin. Right, the chewing is mechanical digestion, and remember what's in our saliva, amylase. The enzyme we worked with in lab that takes a big starch and chops it up into smaller sugars. So all that's going on in the mouth as the saliva mixes with the food, gets a little bit more liquidy, right? That's why we call it a bolus. Swallow the bolus. It pushes that epiglottis down to cover the trachea, so it's routed into the esophagus, the next major organ. Really what we looked at with the esophagus is the structure, the fact that there's a little doorway at the bottom of the esophagus that the food, the bolus, has to pass through. Remember, that's called the lower gastroesophageal sphincter. And once we've passed through that sphincter, we've entered the stomach. Big things going on in the stomach. Maybe I'll just, my battery's about to die. What we hopefully got from the stomach is that both kinds of digestion are continuing. All the muscular layers of the stomach and all those little bumps on the inside called rugae, that is continuing to mash the food up and break it into smaller and smaller pieces. At the same time, we've introduced a new enzyme called pepsin. Remember, pepsin does what amylase does to starch, but pepsin does it to protein, bless you. So pepsin takes a huge protein and chops it up into smaller chunks, much like amylase did for starch. Remember, there's a bunch of acid in the stomach as well that would help pepsin be able to do that job because a lot of acid causes proteins to unfold or denature, and then pepsin can start to chop them up. Again, at this point, we've added a bunch of more liquid. With the stomach acid and the pepsin, it's even more liquidy than it used to be, so we swallowed the bolus. Here's the specific sentence I said last time, right? The bolus enters the stomach through that lower gastroesophageal sphincter, but while it's in the stomach, the bolus is converted to chyme. So bolus comes in, but chyme leaves, and it leaves through the pyloric sphincter, the little passageway at the bottom of the stomach. Any questions on the parts of this system we covered last week before we talk about what happens next? Okay, so I think I talked a little bit about the small intestine, but I'm just going to pick it up like I didn't, I guess, to bring us really back into it. The small intestine is a really important organ in our digestive system because there are two very important things going on here. Number one, we're going to complete, as in finish, the process of digestion that we started in the mouth as soon as we took a bite of food. We're going to talk specifically how that happens with some help from those accessories that I mentioned last week. Remember, accessory organs, the food doesn't ever pass through them, but they're contributing to the digestive process in some way. And I'll add these in just a second. As we finish breaking our food molecules up into smaller pieces, this is where we can actually use them. We can take them into our system. Remember, all of this digestion that we've seen so far happens extracellularly. All this is going on in like the middle of the stomach, the middle of the small intestine, because as you guys know, it's kind of hard for our cells to take up a big bulky molecule. So we're doing all the work in the middle of the organ, trying to chop things up into teeny tiny little pieces like an individual glucose or an individual amino acid. And once we have the little pieces, those are a lot easier to bring into our cells, and that's how they're brought in. Once we chop them up, they're going to be absorbed by the cells that are lining the small intestine, and we'll pass them into the bloodstream, where of course they can be circulated all over the body. Okay? Again, we're going to need some help to do all of this job, to really do the digesting part of this. You're going to hear and see in the video I'm going to show you in several minutes, because the small intestine is so big, so long, we divide it up into like 3 different segments. The only one of those special names I want you to know is this first part. It's called the duodenum. The other ones you don't have to worry about, okay? Why I think it's important to know the first segment is this is where we're essentially mixing in everything we need to do these jobs, and that mixture then travels that whole 6-meter length together. But we got to dump in everything we need right up front for all this to go down. What are we dumping in? Well, our partially digested food, the chyme that's coming into the small intestine from the stomach. Remember, the chyme leaves the stomach through that pyloric sphincter at the bottom, and it's getting dumped directly into the duodenum. So great, we've got our food. What do we need to break it down? Well, we need some more enzymes. Some of these will be familiar, some are ones we haven't heard about before. Most of them, not all, but most of them are actually coming from the pancreas, the first accessory organ I've really mentioned. So the pancreas does a lot of important things for our bodies, but it is playing a critically important role in our digestive process. Even if you had the healthiest diet in the entire world, if your pancreas was not making these enzymes, or not able to dump them into your small intestine, you would be chronically malnourished because your body would be unable to break things down to a small enough degree that your cells could actually soak up those nutrients. So I can't stress enough, I think, how important these secretions from the pancreas are. And they're coming from the pancreas, being dumped right into the duodenum along with the chyme from the stomach. So we've dumped the food in there, and now we're hitting it with all these enzymes that are basically going to chop it up. You guys know how they're going to do that, a bunch of hydrolysis reactions or a bunch of degradation reactions that we've talked about before. The other stuff we're going to dump into the duodenum along with the chyme, told you it was coming, along with the chyme from the stomach, along with the secretions from the pancreas. We're also going to see how the liver fits into the mix here. The liver is dumping in a substance called bile. Bile, I know we haven't talked about yet, I guess until now. Bile is a substance that's made in the liver, and why I have liver / gallbladder, bile is made in the liver and can either go directly into the duodenum or it can be stored in the gallbladder. The gallbladder is kind of like just a little storage pouch for bile. So if you have a bunch of bile, you can just route some of it into the gallbladder and hang on to it till you need it later and then put it into the duodenum. This word emulsify, I'm going to talk a little bit more about later in case you're not familiar with what that means, but I want to point out the word fat right here. This is the first time I've talked about fat in the food that we eat being digested, right? We started digesting carbohydrates in the mouth with amylase as soon as we started chewing. We started digesting proteins in the stomach with pepsin. We don't touch the fats or oils in our food until they're all the way into the small intestine. They're a little bit harder for us to deal with because, as you guys know, they're non-polar. They're hydrophobic. And we've been adding water with saliva and stomach acid and all of that. We've got a bunch of water in with our partially digested food. And so things that are non-polar are just a little bit more difficult for us to deal with. Doesn't mean we can't deal with them, we do. It just takes a couple extra steps essentially. So we haven't touched them until we get into the duodenum, and then we have the bile from the liver coming to help us do this. We're gonna look at what actually is happening, how the bile helps with that once we get specifically to the accessory organs in a couple minutes. I'm going to first talk about the major organs and then we'll kind of backtrack and talk about the accessories before we pull it all together. So I want to talk a little bit more about the small intestine because I feel like, all right, I've thrown out there that we're going to get all these enzymes to keep breaking down our food, which is the first important job happening there. But remember what else is going on in the small intestine? Finally, we're able to actually absorb the nutrient molecules from our food. This is the whole point, right? We're heterotrophs. We have to get our food from a different source. And all we've done up until now is take the big food molecules and chop them down into smaller pieces so that we can bring them into our system. How we do that is what really is being addressed on this slide. And what I want to do here is actually break away from the slide for just a second and have you guys think about a topic that we went over in lab a couple weeks ago. The whole idea of the surface area to volume ratio, right? This was in lab 7 with the little potato cubes. We went over the fact that it's not how much surface area there is, it's not how much volume there is, it's really the ratio between the two. How we thought about it in the context of that lab was which potato cube could suck up the most iodine, or which cells could be the most efficient at absorbing molecules like nutrients perhaps. So we're going to take that same basic idea and put it in this context now. We already know the small intestine is very long, but it's a pretty narrow tube, right? So if we're not going to like calculate the volume of cylinder or anything, but if you thought about a long tube 6 meters long that was this big around or whatever, there'd be basically a set amount of volume, space in the middle, associated with it. So that is whatever it is. So if we want to play with this idea of surface area to volume ratio to increase the efficiency of transport, volume is what it is. The only thing we can play with to increase the odds essentially in our favor is the surface area. And that is absolutely what we have done in our bodies. We essentially find any way, shape, or form we can to pack more and more and more surface area into our small intestine. Remember, surface area to volume ratio. If you can pack more surface area into that space, it increases the ratio, which increases efficiency of absorption. So how have we been able to do this? That's what I'm hoping this figure can help us recognize, okay? So this is a part of a little piece of that 6 meters of length that we have for our small intestine. And lumen, I don't think I've talked about this word with you guys yet. Lumen just means like whatever's inside the tube. It's a term very frequently used to describe organs. So the esophagus, the lumen was where the bolus was passing through. The lumen of the stomach is where the chyme was being formed as the pepsin and acid was interacting with the bolus. The lumen of the small intestine is where our partially digested food is passing. The lumen of a blood vessel is where your blood cells are flowing through it. Lumen just means the inside of a tube or organ of some kind. And the first thing I want to point out is notice this is not perfectly smooth on the inside like they've drawn it on the outside. In fact, this is the first zoom in we're taking is on a little part of the inside of that tube. So what I want you to imagine is imagine this was like a PVC pipe, perfectly smooth on the inside and the outside. So if this were perfectly smooth on the inside, imagine I've got a piece of string and I'm just going to pull it straight across whatever the inside of the tube is. So my string, if it were straight, would be this long. And I like that visual of the string because now look at it with all the little bumps it has. If I were to take a piece of string and trace every last little nook and cranny, all these ruffles and bumps along the way, can you imagine how much longer this string would be if I were to pull it straight? It's at least 3 or 4 times as long, at least, as the one that just went from here to here. So this is the first way I connect with the fact that adding all these little bumps along the way dramatically increases the surface area. And again, the whole point here is more surface area, higher surface area to volume ratio, more efficient transport. What are we transporting? Food into our bodies, into our cells, okay? If we zoom in again, now one of these little blebs over here, this is a structure called a villus. And what you're seeing with this structure called a villus, these little kind of boxes around the outside, those are the actual cells lining our small intestine. So like this up here would be the lumen where the digested food is floating by. And it's these cells right here that are really tasked with sucking up those nutrients. And they're not going to stay with those cells, right? Those intestinal cells are essentially a conduit. They're a way for us to take the nutrients into our system. But look what's right here in the middle. The red and the blue, I know we haven't really seen this yet. We're going to talk more about it in the rat lab. The red and the blue, those represent blood vessels. So I mentioned the circulatory system pretty briefly last week, but remember what I said it's doing? That's how we transport stuff all over our body. So these cells lining the small intestine, they're the ones in our body that are sucking up all the, for example, glucose, the sugar we get from the food that we eat. But you guys know those are not the only cells in our bodies that need that sugar. We need that glucose in every cell in our body to use during cellular respiration. So how do we get it from where we're taking it in to everywhere else? These cells take it in, they pass it right into the bloodstream, and the blood can then circulate it all over the body. This is what's happening with most of our nutrients. Sugars like glucose and amino acids, which you guys know make up proteins. Once we've chopped the big food molecules down into these little pieces, those little pieces are sucked up by these cells and passed right into the blood. Remember how I said fats are a little bit trickier to deal with? We treat them differently. We didn't do anything with them until the small intestine. We're going to see what we do with bile, and some of the enzymes from the pancreas will help us here as well. But once we've broken them down as much as we can at this point, they'll also be, the fat products essentially will also be sucked up by these cells, but they do not go right into the bloodstream. Again, they're nonpolar, they're hydrophobic, they're a little bit more difficult to deal with. And so there are some extra steps we have to take to do that. And the big difference we can see initially is that we don't send them into the bloodstream. We don't. We send them somewhere different. This little white line right in the middle, that's a structure called a lacteal. Okay? So sugar products and protein products right into the blood. Fat breakdown products go into the lacteal. The lacteal is a vessel, kind of like a blood vessel, but it's a little bit different. It's part of our lymphatic system. And this I'm going to take maybe a minute and explain because I usually would have explained it when we talked about cancer in Chapter 9. But since we skipped that entire part of Chapter 9, you guys haven't heard me talk about this system. So the lymphatic system, much like our circulatory system, right, all our blood vessels, the lymphatic system is also made up of a whole bunch of vessels that go all over the body. But there's not blood in the normal sense in there. There's a liquid called lymph. White blood cells, which are part of our immune system, also travel there. And then it's also helping us absorb fat from the food that we eat. So in terms of what do you need to know about it for this class, you just need to know it's where fats are sent. In later steps that we're not looking at, that fat would eventually be mixed in with the normal blood supply and be able to go all over the body. We just have to process it differently. And the only aspect of that different processing that we're looking at is the fact that it comes into the lacteal, which is a specific type of lymphatic vessel. That's all you need to know about it. Okay, so it's what's written right here on the slide. You're good. Any questions about what's happening in the small intestine? The fact that we're going to finish digesting all those big food molecules from the food that we ate and using these cells in the small intestine finally soak up those nutrients so we can get them all over the body and use them. Okay, so we have one more major organ to talk about, and that is the large intestine. Again, the name here is based on diameter and not length. This one's actually much shorter than the small intestine, but it's bigger around, hence the name. This one, even though it's shorter, is also divvied up into 4 different segments. The cecum, which is the very first part of it. So from the small intestine, our digested food, really the remnants of our digested food, whatever we couldn't use essentially, gets dumped into the cecum. On us, that's where our appendix is. We'll see a little picture of that in just a second. The appendix looks like a little pouch kind of hanging off the cecum. Our rats that we're dissecting, they do not have a dedicated appendix. I think that's like a question in the lab book, and I wanted to mention it before I forgot. I'll say it again in lab. Rats do not have an appendix like we do, or like some of us do if you still have yours. From there, we go into the colon, which itself is divided into different segments. You don't have to know them, but I'm going to say them right now, only because you're going to hear them in the video in a minute. The 4 parts of the colon are named based on kind of what they look like or where they're going. The first part, you're going to start low, like on the side of your body. The first part is called the ascending colon because it moves back up. Transverse cuts across to the other side of your body. The descending colon goes back down, and right at the end there's like a little squiggle called the sigmoid colon. Okay, so colon has, again, you don't have to know this, just mentioning it so you hear it before the video. Ascending, transverse, descending, sigmoid colon. It's all colon, okay? From there, we'll store whatever waste we still got in the rectum before we eliminate it from the body. Functions. What is the large intestine really all about? Number 1, we're gonna absorb some stuff, okay? What are we absorbing? Well, remember, we've added quite a bit of liquid to our food as we've digested it. Initially saliva, then the stomach acid and secretions, then all those secretions from the pancreas and the liver during the small intestine. We've added quite a bit of liquid to it. We're going to reabsorb some of it. Okay? We're also going to soak up some salts and some different vitamins before we store the waste until we can get rid of it. The big point I always make here for the large intestine, do you see the word digestion anywhere on that list of functions? No. Remember, that's done. We finished whatever we could do in the small intestine, the last organ. Now that we're here, we're not digesting anything. We are not digesting anything. There are some things there that are doing some digestion, but we're not a part of it. Okay, it's just happening in this organ. That sounds weird for me to say, right? Like, who the heck is there that's digesting stuff if we're not? That's what I want to talk to you guys about, at least for a couple minutes. The large community of bacteria that live inside our large intestine, because they're really important, as it turns out, to our health. Before we talk about the bacteria, any questions on what we are doing in our large intestine? Okay, so what do I mean by large community? Millions upon millions, billions probably upon billions of bacteria live in our large intestine. Here's that little flap. Before I forget, this little pouch, that's what our appendix kind of looks like. So here's showing you the small intestine as it would be dumping into the cecum, the first part of the large intestine. And this is where there are a whole bunch of bacteria. Not just in the cecum, they're really throughout. Sometimes it freaks people out to know that there are billions of bacteria living inside you. Especially if you're any bit of a germaphobe, it just sounds creepy a little bit. And I want to caution you against being totally freaked out by this, because I feel like a lot of people have this negative connotation with bacteria because they can make you sick. In all honesty, it's rare that they can make us sick. It just so happens that the ones that make us sick are the ones we always hear about. So all bacteria kind of get a bad rap based on some of the bad guys. Most bacteria can't make us sick, and we're learning more and more over the last 10, 15, 20 years about the ones that actually interact with our bodies in a really important way to help us stay healthy. Bacteria at this point have been linked to everything from allergies and asthma to perhaps obesity and certain mental health disorders. Like, this is a really active area of research, and new findings are being discovered all the time. So I want to talk about specifically what's happening with digestion, because that's what we're focused on, of course, right? Even though these bacteria may be involved in other things, we want to think about digestion because that's what the chapter's about. So what are they doing in terms of digestion? Well, whatever we can't use, whatever has essentially passed right through us and winds up here, that becomes food for the bacteria that live here. That's sort of what they get out of this. They get a place to live and they get food. Whatever food we couldn't eat goes right through to them. But we get something out of it too. And that's what I'm really trying to get at with these last two things on the slide. They produce certain vitamins that we lack the ability to make on our own. Great example is vitamin K. Now we clearly have, bless you, we clearly have not talked about different vitamins or what they do or why we need them or whatever, but vitamin K is really important for blood clotting. Like if you have an injury and you're bleeding, even just like a little paper cut, why are you bleeding? Because there's been damage to some blood vessels and blood cells are spilling out of that blood vessel. If we want to stop bleeding, which is important, we have to be able to blood clot. And vitamin K, blood clotting is really a complex process, but vitamin K is really important for it. And we cannot make it on our own. These bacteria make it while they're living in our large intestine and we absorb it from them. They make it, we say thank you very much, we soak it up and we can use it. And what I usually always talk about here at this point is if anyone has kids, I don't know, usually right when a baby is born, one of the first things that happens within the first couple hours of their life is they're given a shot of vitamin K. And sometimes people are a little bit freaked out by that, like my perfect brand new baby, like why are you giving them a shot? Leave them alone. It's a really important shot to get. Why? When we're born, we're sort of a clean slate here. Like you're not born with a huge population or community of bacteria. That community only really becomes established in the first minutes, hours, days, weeks, months of your life as you're exposed to the outside world. And so until that community gets up and running, you have no one to make your vitamin K for you, meaning you would have a real hard time clotting blood to recover from any injury. So can you imagine if there's a baby who can't make vitamin K, who doesn't have bacteria making it for them, and they have some kind of injury, internal or otherwise, that baby could essentially bleed out and die. This is why that shot of vitamin K is so important. It gives the baby enough vitamin K to essentially buy them time until their bacteria can start making it for them, right? So when you understand like why it's happening, giving that shot to a little baby doesn't sound so bad anymore because it could literally save that baby's life. Honestly. The other thing, again, as a microbiologist, I love to talk about that these like good guy bacteria that are supposed to be there, they can actually help protect us against the bad guys, the pathogens that can make us sick. And this is going to be a vast oversimplification, but just as one way of thinking about it, it's kind of like a real estate issue, right? If this space that's here is all occupied by the good guys and one bad guy gets in there from like eating food that's contaminated or whatever, there's nowhere for them to like settle in and set up shop, right? So it's less likely for you to have those issues if you have a thriving community of the good guys, which is something I always mention at least a little bit. So any questions on the fact that you have a whole bunch of bacteria in your large intestine and why that actually is very useful for us as a species? Okay. So we've heard about all of the major organs and what's happening there, right? Stomach, let me start with the mouth. Mouth, stomach, and small intestine, we're digesting. Once we break everything down in the small intestine, we absorb whatever nutrients we can use. Whatever we can't goes right through the large intestine, where maybe the bacteria can break down some of it. We're absorbing water, we're absorbing salt, we're absorbing vitamins, and the rest is just being stored until we can get rid of it. What I want to do at this point is double back to those accessory organs I mentioned a little bit a few minutes ago and make sure we appreciate what is going on with them. So what we're going to do is here on 3 separate slides about each of the accessory organs, then we'll pull it all together by watching a summary video, and then kind of overview of the rest of the chapter, I guess. And then what I want to do is really talk details about chemical digestion. Because we've heard like a little bit here and a little bit there and all different organs and all different enzymes. And I want to make sure we have a full understanding and complete picture of how the different molecules in our foods that we can use are actually broken down. And we have to talk about the pancreas before we can do that, because as I mentioned earlier, the pancreas is making a lot of really important enzymes that help us or really allow us to digest our food completely. So, you saw in the overview picture last week, we'll see another image in just a minute to show you where the pancreas is. You will be finding it in your rats. I think it's actually one of the harder organs to find in the rats, so I'll help you guys find it, obviously. We're not going to worry so much about this endocrine and exocrine function deal. In chapter 31, I usually talk about glands and like what's an endocrine and what's an exocrine. We haven't done that, obviously, right? So what we're going to focus on, you can just cross out endocrine if you want. If you know someone who's diabetic, you may already be aware the pancreas is related to diabetes. I can tell you more about that if you're curious. We're not going to worry about it for class, okay? We're focused on what's going on down here under exocrine. You don't have to know what exocrine means, but in case you're curious, exocrine glands are glands that make up some kind of product like an enzyme and secrete it into some other organ or some body cavity or whatever. And so what the pancreas is making that we're paying attention to for the digestive system is this really, delicious sounding pancreatic juice. What the heck is that? It's just a mixture of all the different stuff the pancreas makes that it's sending into the duodenum. You guys remember what the duodenum is? It's that first part of the small intestine. Okay, great. So what's in this juice that's being sent there? First component, sodium bicarbonate. Okay, write that down if you don't know this, sodium bicarbonate. That should maybe sound vaguely familiar because we actually used that in lab a couple weeks ago. Why it's relevant here is a property that I never even once mentioned to you in lab. Sodium bicarbonate is basic, like as in the opposite of acidic, okay? It's a basic molecule. Why might that be relevant? Let's think about the duodenum, right? From the stomach is coming the chyme, right? What was the stomach acid? 2, right? pH 2. And so what we're doing is we're immediately hitting that stomach acid with a base to neutralize it. It was very acidic, you add the base, and bam, we wind up right in the middle back to neutral or slightly even basic. I'm making a big deal out of this because all the enzymes that the pancreas makes that help us digest our food, they work best at neutral pH. So we have to neutralize the stomach acid with the sodium bicarbonate to make the intestine, the small intestine, have a neutral pH and not be acidic like the stomach was. So what I've got listed here for you are 3 different enzymes that are helping us digest our food. All of these are made by the pancreas, mixed up with that sodium bicarbonate and delivered right into the duodenum so they can mix together with the food, the chyme, coming from the stomach. Here's one you already know, right? Amylase. You know what this does. You knew it from lab 8. You knew it from the mouth where it was already in the saliva. Here's where we make more of it. You already know what it's doing. Takes a big starch molecule and chops it up into smaller pieces. Why in the world are we making it again if we already made it up here? Right? Let's kind of track what has happened with the amylase. It's made in saliva, and as you're chewing, which you don't chew your food for all that long normally, right? As you're chewing, we start breaking up starch, but then you swallow it, and where does it go? Your stomach. What's the pH in the stomach? 2, right? It's acidic. You guys tested amylase at pH 4 in the lab, and it did not work, right? So as soon as that amylase gets to your stomach, not going to work anymore. So at that point, we've only partially digested whatever starch was in the food that we ate. We got to finish the job. That's why the pancreas is making more amylase that will come into play in the small intestine and finish chopping up whatever starch we hadn't done yet. Likewise, second enzyme we've seen that can digest proteins. Remember we saw pepsin in the stomach that took a protein and started chopping it up. Pepsin works best at pH 2. But once that chyme passes into the small intestine, are we at pH 2 anymore? No. So again, as soon as the pepsin, like the chyme, gets to the small intestine, the pepsin can't work anymore. And so we have to bring in someone else to pick up the slack. Essentially, pepsin and trypsin do virtually the same job. They take a big protein and break it up into smaller pieces. It's like they're passing the baton. Pepsin did what it could in the stomach and then says, here you go, trypsin, you finish what I started. So we're seeing carbohydrates being digested. We're seeing proteins being digested. Remember, we started this in the mouth. We started this in the stomach. This is the first time we're seeing an enzyme that is used to break down lipids. It's called lipase, right? ASE is a hint that it's an enzyme. Lip for lipid. So it's called lipase. Lipase breaks down fats or triglycerides. So I'll give you like a reminder later about the structure of fats and oils once we're looking at chemical digestion. This enzyme helps us break apart big fat molecules. And again, the pancreas is making all of these enzymes and dumping them into the duodenum where they can mix with the chyme and help us digest our food. Any questions on the pancreas? All right. Next up, liver. Liver does a whole lot of important stuff. The only thing you have to know, the only thing you need to write down is the second bullet point. Produces and secretes bile. That's it. We're not going to talk about all this other super important stuff that the liver does, which is a disservice to the liver, I suppose, to not talk about, but we're going to focus on the digestion aspect that we're focused on for the whole chapter. This is where bile, which is that substance I mentioned earlier, helps us digest fat. This is what makes the bile. It makes it, and it can either secrete it directly into the duodenum, or we can put it in that little storage pouch, the gallbladder, which we'll talk about on the next slide. This is probably the first organ you're gonna identify in your rat, because it's in them as in us. It's a pretty big organ. It's the first thing you'll notice, I'm sure, once we get to that point on Wednesday. Any questions on how the big thing you need to know about the liver is that it's making bile and putting it into the duodenum. Yeah, add that because I didn't write that here. Add it. Secretes bile into duodenum to remind you where it's going. Okay, so we might go duodenum, the first part of the small intestine. Yeah. Okay, so sometimes we go directly into the duodenum from the liver. Sometimes, like I said, we make a stop in the gallbladder, okay? Once again, we're not going to talk about all of this, so I'm not going to talk about, gallstones or the fact that some people have their gallbladders taken out. Major function you need to know, storage of bile. It's like a little Tupperware for bile. If you got a whole bunch produced and you don't need it all right now, put it in the gallbladder to save it till you need it. That's it. That's it, okay? So, our three accessories, pancreas, liver, and gallbladder. Pancreas and liver are making some really important products for us to actually be able to do digestion in the small intestine. And the gallbladder just helps us store one of them if we need to. What we're going to get on the next slide is just a picture of where all these accessory organs are in relationship to the major organs we've seen so far. So I like the figure on the next slide because it just gives us a quick frame of reference. What we're missing here is the bottom of the stomach. Okay, so I'm going to show you where it would be because I think that can help situate us. Remember how the stomach's kind of J-shaped? The bottom of that J would be nestled right in here. I think they left it off to have room to write all these words. The bottom of the stomach would be here. Remember, the stomach dumps the food into the duodenum. So this is that first part of the small intestine called the duodenum. And I think that, to me, makes perfect sense to look at where these accessory organs are because they're all situated pretty closely to where we need them to be to effectively and efficiently dump stuff into the duodenum. So we would have the chyme coming in from this way. The pancreas is really kind of sandwiched right up in that little, there's like a little nook you'll see when we get our rat open where the stomach and the small intestine come together, and the rat, it kind of looks like a little triangle, and the pancreas, or part of the pancreas, sits right up in that little opening right there. And that gives us the perfect way to dump in that, I always say, delicious sounding pancreatic juice. Remember what that has? The sodium bicarbonate to neutralize the stomach acid, and the mixture of all those enzymes that help us digest our food. The lipase, the amylase, the trypsin that we just talked about. This up here is part of, not the whole thing, part of the liver we're being shown. Remember, liver makes bile, which can either get dumped right in to the duodenum, where the food is going to be waiting for it, or we can route it into the gallbladder, a little Tupperware, a little storage pouch, and then get dumped in later as needed. So again, I want you to know the term duodenum, because this is where everything we need to digest our food is essentially being mixed together, and that whole mixture will travel that 6-meter length of the small intestine. That's where we will finish digesting our food and absorb the nutrients contained within it. Any questions on these accessories, what they're doing, where they are? All right, so let's bring it all together by watching this video. That looks weird to me. I'm going to open it on the other page. So this video is posted, obviously. I just clicked on the linked out one, which I'm sure I could have resized, but I guess I'm being lazy. And I am going to open it on this page because I know it will play on this computer. This video, I think, does a pretty good job of taking us through all the parts of the system that we've talked about. There are going to be some words mentioned that you don't have to know. Remember, small intestine has 3 parts. The only part you need to know is the only one I've told you about, duodenum. They're going to mention the names of the other parts. They're also going to tick off the names of all the parts of the colon that I mentioned to you. Again, I didn't have those written down anywhere. You don't have to know them. Of course, after the video, I'll see if you guys have questions.
The primary functions of the digestive system are the breakdown of food, called digestion, and absorption of nutrients. Digestion begins in the mouth, where the teeth break food into smaller particles during mastication.
Chewing.
Salivary glands located near the oral cavity secrete saliva, which begins chemical digestion and keeps the food moist. As food is swallowed, the soft palate blocks the upper pharynx to prevent food from entering the nasal cavity, and multiple voluntary muscles in the face, neck, and tongue contract, pushing food particles through the pharynx. The food passes over the epiglottis, which prevents food entry into the respiratory system, and then into the esophagus, which connects the pharynx to the stomach. The one-way movement of the food mass, now called a bolus, is controlled by wave-like involuntary muscle contractions. This movement is known as peristalsis. The bolus now enters the stomach.
I'm going to pause it real quick. They didn't name it, but I want to remind you how it enters the stomach, through that lower gastroesophageal sphincter, right? There's a muscle wrapped around the bottom of the esophagus that's usually closed, and when the food is coming, it like knocks on the door. That muscle, the sphincter, relaxes, and then the food is allowed to pass in.
Folds in the stomach wall, called rugae, allow for expansion as the stomach fills. Stomach cells secrete hydrochloric acid, pepsinogen, and various regulatory hormones that chemically digest the bolus. Muscular contractions in the stomach churn its contents to further break down the bolus and mix it with stomach secretions to form a thick liquid called chyme. Chyme exits the stomach through the pyloric sphincter and enters the small intestine, the major site of nutrient absorption. The small intestine consists of 3 parts: the duodenum, jejunum, and ileum. Bile from the liver and digestive enzymes from the pancreas empty into the duodenum to aid in digestion. Absorbed nutrients pass from the lumen of the small intestine into blood and lymph. Chyme not absorbed in the small intestine enters the large intestine. As it passes through the cecum and ascending, transverse, descending, and sigmoid colon, water and salts are absorbed and chyme is converted into feces. The rectum stores feces until nervous stimulation initiates the defecation reflex, resulting in elimination through the anal canal.
All right, any questions about the video or any questions about anything we've discussed with the digestive system? Okay, so I'm going to say let's go ahead and take our break. It's 10:51, be back at 11. We've got 3 slides left in this chapter where we're going to look at all the details of chemical digestion for each type of nutrient molecule: carbohydrates, proteins, and lipids. All right, so be back at 11. Yes, do you have them or you want me to just show you? I'll just show you. There's quite a bit. We skipped a lot.
You did? Oh,
Yes,
sweet.
so I know, it's good news for most people, right? Yeah.
No, yeah, I did not do anything over break. You were right.
Yeah, I know, I'm assuming if you're
We
somewhere
were
and so busy, yeah.
Oh my god, it was unbelievable. Like,
Yeah. Did you have fun though?
yeah, we did
That's
really
good.
good actually.
That's good. Was it like a tournament or?
Mhm.
Yeah, cool. All right, so let me pull up.
So I know we skipped the cancer slides.
We
I heard
skipped
you
all
say
the
that.
cancer stuff, yeah. So let me pull up all the lectures
Okay.
and we'll. Okay, so chapter
And
9.
Are you asking for both parts of the rat lab, like part A and B, for the lab quiz?
Oh, I don't even know if I said this to you guys. It's a really long lab. So for the pre-quiz, focus on the intro and then like flip through and see what else we're gonna look at, like organs, blood vessels, whatever, but
Just
I'm
the
not
first page.
asking detailed questions
Oh,
about
okay.
stuff. Yeah, because that's like the point of the lab is we're gonna figure all that stuff out.
Okay, cool.
Yeah. All right, so from chapter 9, we skipped structure of eukaryotic chromosomes.
Okay, wait. So chapter 9.
These slide numbers are different than the ones.
Oh, they are? Okay,
Yeah.
so the structure of chromosomes.
Structure of chromosomes and the picture that follows it. And then all the cancer stuff, which is about 7 slides, 8 slides.
And the 7 slides.
I think it's 8. 1, 2, 3, 4, 5, 6, 7, 8. Yeah, 8 cancer slides.
Slides that has cancer.
Yep.
That's cool.
Yeah. And we did probably more in chapter 31 we skipped. All right, in chapter 31.
When you said chapter
oops.
31, I'm like,
You're like, what
did
the
I
hell?
miss?
What chapter? Yeah.
I was only
This
gone for
is
a
an
week.
older version. Okay, let me see. Chapter 31. Oops. That's not the one I want. Sorry.
Hey, it's cool.
Okay. We skipped. What I would actually say is start chapter on from tissues to organ slide. So we
Wait,
skipped
from tissues to organ slides.
we skipped like the first 17, 18 slides of that. Gone.
Okay.
You start there and it's then the whole part of the
Exactly
chapter
that.
read is like 10 slides.
Oh.
That's it.
Oh, okay, cool.
Okay. And then in chapter 34, which we're doing right now, we're going to keep all of it essentially.
Keeping all of it.
Yep. And then you'll see the rest that we cross out in chapter 10 and chapter 42.
Awesome.
So that
Thank
cuts back
you.
significantly. Yeah. Hey,
So
I
was a
saw your
little-
emails. Yeah, sorry.
and
I
no-
didn't respond
no.
because I saw your second one. I was
Yeah,
like, okay.
Please don't respond because I'm done. And just
No, no,
totally,
no,
like,
no, no.
Didn't scroll down far enough.
No, no, no.
So I haven't done a works cited in like 15 years.
Yes, no,
So
and I feel like I'm very
if it's cited
loose about it. If it's
okay.
cited and you attempt to put it in some kind of, you don't just paste like www
yeah,
dot
no,
whatever.
I
You're
used
fine.
the thing.
Yeah, yeah.
I guess that was one of my questions is
Yeah.
does it need to be in like alphabetical order or anything? Okay, cool. Then, so when we were doing the like relationship to class concept,
yeah.
is that, should I like write like chapter, put like in parentheses like chapter 24,
Yes,
lab
yes.
5,
So like
or-
that is really all that.
and then
this,
cite it again at the bottom?
Yeah, all this section is literally like a sentence. Like let's say you're doing an article about photosynthesis, whatever.
I'm doing, it's gut seeding using
Cool, yeah.
genetically modified bacteria.
That's really cool.
Yeah, so it relates to like the lactic acid fermentation
Yeah,
lab. It relates
and
to
digestion.
this.
Yeah, so you could just say like this article relates to the digestive system from chapter 34.
Okay.
That's this entire section.
I'm,
Is
like,
that one
overthinking
sentence?
it.
Yeah.
I wrote like
What-
6 pages. And
well,
then I was
probably,
like, oh,
yeah.
it needs to be double spaced. I
Probably-
can cut this out.
Probably the rest of what you wrote there. If you're
Discussion.
talking about like how it like what does it actually relate to in the digestive system, that's what would go in your discussion.
Okay,
yeah.
cool.
Yeah.
I think it's already done. I'm just,
Yeah, probably. Yeah. Like I said, I really designed it to not be like a huge pain in everyone's ass.
Yeah.
Just like a way to find something out in the world that you can understand better because of what we've done in class.
Definitely.
Yeah.
Thank you.
Cool. Yeah. Hey,
On this one right
Yeah.
here, it's 4, then is it 4 again?
it's not because remember, as soon as they're split,
So-
they count as separate.
Separate, okay.
So that one would have-
It'd be doubled.
Yeah, it'd
then
be
up-
8.
Okay.
Yep.
those
Good.
would be-
Yep.
31st.
Okay,
So
okay.
we have lots of time.
Gotcha, yeah, because I walked in like right towards the end. I walked in, she's like, oh, okay.
Yeah, what I totally misread too is I misread that if you do not turn in all the CTF assignments, you will not be eligible for credit. And I read this if you turned it in, then you ended up with it. Doesn't make any sense. It's like, get 30 points, I want the 30 points.
Okay, that's good.
Yeah. We are. We start today.
Oh, do we?
Yeah.
Oh, is it?
It's in 3 days.
Oh, wow.
Yeah, it's today and Wednesday and next Monday.
Wow.
It's a big lab. Look, there's so much information.
I didn't get a check. I didn't actually so I have to review the diagram and watch the video.
Yes. Did you get this thing?
Yeah.
Cool.
Yeah.
Do you want first? If it's a group thing?
Yeah, sure.
Cool.
Yeah, I got mine ready. It's on
Yeah.
so.
I've been trying to like study it a little bit so that it's, and this is really, which I didn't realize that this is like a guide.
Yeah.
If you can't find it, do this. I was like,
Yeah, it was really helpful.
it's gonna be interesting, because there is definitely, like, I'm not really good with, veins and arteries, so the circulatory system.
Yeah.
I'm like, I feel pretty confident with about every verse, like 7 out of 10 or whatever that is, but all of this, I'm like,
Yeah, it's
I know that that's the jugular, but like,
Yeah.
A parent cell will be separate to daughter cell.
Yeah, for mitosis, they're the same.
That is
They're the same, right? So with the cell cycle or with mitosis, whatever you start with, that is exactly the 2 cells you're making.
So after making in total have 3 or 2 cells.
2. You take your one parent and you split it in half. So those 2 daughters are identical. They're the same as each other. They're the same as whatever the parent was. Yeah. Yep.
Yeah.
since she said we weren't opening them up, she said it's gonna do that Wednesday.
Yeah.
I think it's today is gonna be getting everybody past the date factor, like handling something that, I'm not very used to encapsulate placenta still living, so like I'm really not.
Yeah, so it's not.
No, this is fine, as long as I got gloves on, I'm cool. I was like, can I be the weird person that has like, this is the red one, this is the red one. I would do that, I'm not very
Okay, guys, we are going to get back into it. What we have left to do in chapter 34 is, like I said before break, we're really just going to look specifically at putting all these little bits and pieces we've heard along the way together so we can look in one slide. What are we doing with carbohydrates in the food that we eat? On the next slide, what are we doing with proteins in the foods that we eat? And on the last slide, what are we doing with fats that are in the foods that we eat? Okay, so why I said we've heard a little bit about this along the way, we heard about amylase in the saliva. We heard about pepsin in the stomach. We heard about all those things being made by the liver and the pancreas. Like, how do we really put that together in a meaningful way to track what we actually do with each type of food molecule? Those 3 kinds of molecules, carbohydrates, proteins, and lipids, you'll sometimes hear those described as being macronutrients, because those are the big kinds of food molecules we can use for energy in our body. So if you've ever taken like a nutrition class or anything like that, you've probably heard them described as macronutrients. So what we're going to do is just string together the parts we've heard about along the way and maybe add in a couple new details for each one as well. Okay, so carbohydrates, what do we do with them initially? You already know. We're going to use amylase. You already know what reaction this catalyzes, the hydrolysis or breakdown of starch. You already know how far it takes it, right? You know this from lab. It takes starch and breaks it up into little maltose molecules, which are not labeled up here. Maltose is a disaccharide. This is one molecule of maltose. This is a second molecule of maltose. I'm stressing it's not labeled and stressing maltose because right there it says maltase. Maltase is not the name of a sugar. What does ASE mean? Enzyme. Okay, this is one of the new bits we're going to add in. Before we add in the new bits, I want to remind you, we've already heard that there are 2 different versions of amylase that we make. First one is made in the saliva, so as soon as we start chewing our food, already we're starting to break down the big carbohydrates that are there. Once we get that bolus to our stomach, the stomach acid like null and voids the amylase from the saliva. Once it gets in the acidic condition of the stomach, that amylase is no longer functional. That's why the pancreas makes more for us. We dump it into the duodenum, the small intestine, which is where we're picking up this figure, right? So I wanted to make sure you guys knew where we were for this figure. This is the lumen, the inside of the small intestine. So way up top there, we've got part of a larger starch molecule, and you're being shown amylase, which at this point would have been amylase from the pancreas. That is gonna rip that bigger carbohydrate into maltose molecules. Here's what I said is the new little bit. In the small intestine, there's an enzyme called maltase. What does maltase, the enzyme, do? It breaks down maltose, the sugar, right? Maltose, we very briefly saw way back in chapter 3, but we didn't, focus on it a whole lot. It's just made of 2 glucose molecules put together. So what the enzyme maltase does, picture a little pair of scissors, it just clips the connection between them. Maltase, in other words, catalyzes the hydrolysis of maltose into 2 molecules of glucose. You guys are very familiar with glucose at this point. It's our favorite form of sugar, smaller, easier to absorb. So once we've gone from having a massive starch molecule made of hundreds of glucose molecules to these smaller maltose molecules, ultimately to glucose, those are the little itty bitty pieces that these cells can easily absorb and pass right into the blood vessel. Remember, carbohydrate breakdown products like glucose are being passed directly into the bloodstream where they can then be delivered all over the body. I put this little note at the bottom, enzymes and disaccharides. What is that all about? You guys know how specific enzymes are. Maltose is not the only disaccharide that exists. It's not the only one that we eat. Lactose, which we've talked about recently in lab, that you get in any dairy products you eat, lactose is another disaccharide. Do you think that maltase can break down lactose? No. For every different disaccharide, you need a different enzyme. To break down lactose, we use lactase. To break down sucrose, which is like table sugar you put in your coffee or whatever, we need sucrase. There are different enzymes to break down each sugar, and all of those are present in the small intestine. So that once amylase has done its job, we can break down not only maltose that came from the starch, but also any other disaccharide we get from our food that we eat. Whatever sugar it was, we break it down into monosaccharides that are absorbed and passed to the bloodstream. Any questions about how we deal with carbohydrates, how we digest them and absorb them with these cells lining the small intestine? Okay, so on the next slide, we're doing the same thing, but instead of looking, oh yeah, go ahead, Blake.
What did you say it breaks down into?
Individual sugars, so monosaccharides, simple sugars. Next slide, we're staying with the same idea, but instead of looking at sugars like we did here, we're going to look at proteins. Again, what I'm hoping that will help us in this part of the chapter is chapter 3. It has been a while, obviously, since we've done chapter 3, but if you're not remembering, like, what's a monosaccharide, what is this, what is that, go back to chapter 3 and flip through real quick. That's where we learned how to build the big molecules. We're just doing the opposite now, right? Our digestive system is just breaking down the big molecules in the food that we eat so we can absorb the little pieces. Details of how we do that with proteins are on this slide. And again, we're really putting together 2 things we've heard before and adding in like a couple new details. That's it, okay? What have we done before to break down proteins? We've already seen and heard about 2 enzymes that are getting to work to do the chemical digestion of this type of macromolecule. First one we heard about was pepsin. Remember, pepsin is made in the stomach, works beautifully at pH 2, which is where it's got to work anyways. What does it do? It takes a big protein and breaks it down into smaller pieces. As soon as that chyme passes from the acidic stomach into the small intestine, where we're picking it up, right? I added the label again so you'd know where we were. This is the middle of the small intestine, and because of that sodium bicarbonate from the pancreas, it's no longer acidic, it's neutral to slightly basic. Pepsin does not work in neutral to slightly basic. That's why the pancreas churned out another version of this enzyme for us. Remember, trypsin, like I said earlier, essentially does the same thing as pepsin, it just works under a different pH range. Pepsin worked great in the stomach, but cannot work in the small intestine. Trypsin works beautifully at the pH that's present in the small intestine. What both pepsin and trypsin do, we're getting a visual on here. So this long chain up top, that represents several amino acids hooked together as part of a big protein. And what pepsin in the stomach would have done, or trypsin here in the small intestine can do, is they break it down into peptides. That does not say amino acid yet, right? Pepsin and trypsin break it down into smaller chunks, maybe 3 to 5, maybe 7 or 10 amino acids long, still stuck together. It's not a whole protein with like 200 amino acids. It's a smaller chunk, but still kind of bigger and bulkier than what these cells really want to deal with. So here comes the new step. In the small intestine, there's a new enzyme we haven't heard till just right now called peptidase. ASE enzyme, what does this enzyme do? It breaks apart the smaller peptides, right? So it takes a little chunk, 3 to 5, 3 to 10, whatever, and it's going to clip the individual bonds that were holding those amino acids together. So peptidase takes us from a peptide to having individual amino acids. Again, once we've got that, what we learned about in chapter 3 is a monomer, one of our building blocks that proteins are made out of. This is as small as we're going to get it. And so our cells can much more easily take up that small little piece. And again, proteins, much like sugars, their breakdown products are passed directly into the bloodstream where they can be circulated and mixed, delivered essentially to all of the cells all over our body. Really try to make the connection here. We take a protein in the food that we eat, chop it up into individual amino acids. The cells lining our small intestine suck them up, and through the blood we deliver those building blocks to our cells. Those cells can then take them up and we can incorporate them into the proteins that we're building in our cell at that moment. Any questions on how proteins are digested? Okay. Last macromolecule we're going to talk about. Last slide, really. There's one summary slide, I guess, but last kind of new slide of information is what do we do with fats? I've said a couple of times already, these are a little bit trickier to deal with. Remember the whole idea that fats and oils are both non-polar. They're hydrophobic. They don't like water. And we've got quite a bit of water in our digestive system, right? We've added in saliva and stomach secretions and pancreatic juice and all of that. So all of these liquid components, they don't really interact very nicely, I'll say, with the nonpolar hydrophobic fats that we eat. This is why they're a little bit trickier to deal with. And in fact, I mentioned earlier, we do nothing with the fat in our food until we are in the small intestine. The first thing we do with these fats is we hit them with the bile that was made by the liver and possibly stored in the gallbladder. Either way, it's getting dumped into the duodenum where it can mix together with the chyme, the partially digested food from the stomach. What I want to talk about is the word emulsify, in case you are not familiar. And what I want to do, hopefully to help, is draw a little picture on the board. I know we know about phospholipids because we've talked about them a lot before. And I want you guys to think about the structure of phospholipids as we've discussed them, right? Hopefully we all remember polar head group, so polar hydrophilic likes water, nonpolar fatty acid tails, so nonpolar hydrophobic doesn't like water. Bile has a somewhat similar structure to phospholipids. So essentially what we're going to do is make a coating around fat molecules. So if there's a fat molecule, this is my very technically drawn fat molecule, okay? If there's a fat, what we do is coat it with bile by pointing the nonpolar hydrophobic parts towards the fat and leaving the hydrophilic parts around the outside. It essentially makes a little coating that can help us deal with the fats a little bit more easily. It breaks them up into smaller, more manageable pieces. That's what emulsifiers do. First-hand experience how you might be familiar with this idea. If you've ever baked anything where the recipe calls for oil or butter and water, you guys know oil and water don't mix, right? So if you added those ingredients first, you'd get the two separate layers. Eggs contain natural emulsifiers. So if you put an egg in there and beat it together, the whole mixture comes together a lot more easily. That's what eggs are doing in baking. It's one of the things they're doing. They bind things together, but they're an emulsifier so that the water and the oil in the recipe can come together and be kind of spread throughout the whatever you're baking more easily. So how you're seeing that in this figure is the bile of those little green dots, and you're shown, you know, it's coating whatever fat molecule, droplet, whatever you've eaten. That's where we're now going to be able, once we've got it kind of locked down in a form we can grab it easier, that's where we're going to see this molecule or this enzyme we heard about from the pancreas called lipase. Remember, we heard about this already when we talked about the pancreas. It's another part of that, you know, delicious pancreatic juice that's secreted into the duodenum, which is once again where we are. We're thinking about what's happening inside the small intestine. The bile first coats the fat. That makes it in a format we can use it easier. And what do? We hit it with lipase. I want to talk to you about what lipase does, how this enzyme works. And to do that, first I've got to remind you, or I want to remind you, about what fats really look like, the structure of fats and oils. So again, we're digging way back to chapter 3 here. Remember, fats and oils are both triglycerides. We had a glycerol molecule with 1, 2, 3 fatty acids attached to it. And what the enzyme lipase really does is it grabs one of the fatty acids and rips it off. It grabs a second fatty acid and rips it off. And so what we've got is a couple of fatty acids that have been pulled free and one fatty acid still attached to the glycerol. That's what a monoglyceride is. Glycerol with one fatty acid. So these chunks, the monoglyceride and the fatty acids, those are still kind of big. They're typically bigger than like 1 glucose. They're typically bigger than 1 amino acid. They're still rather big and bulky for us to deal with. And so again, our body has to process them just a little bit differently. Those, you know, smaller than a whole fat, those smaller pieces are still able to be taken in to these cells lining our small intestine. But once they're in there, it's almost like we put them back together in a way. We package them into a chylomicron, which you, if you want, can think about kind of like a vesicle, like a special kind of vesicle to put these fats into. So we package all these pieces into the chylomicron, and that structure, that entire structure, is essentially passed, this time not into the bloodstream, right? This is the point I made earlier when we were looking at the small intestine. Instead, the chylomicron passes into that lacteal. It was that white stripe in the middle of the villus on the small intestine slide. It's a special kind of lymphatic vessel. Like I said earlier, eventually, after we take a couple other steps that we're not looking at, eventually those fat breakdown products will be mixed in with the general blood supply. So it'll kind of filter back into the blood later, but there's some extra work we have to do first, and we're not going to look at those extra steps. Okay, so everything you have to know about fat digestion is on this slide. And again, it all takes place in the small intestine. We'd hit it with bile from the liver, we hit it with lipase from the pancreas. Those littler chunks are then absorbed, packaged into a chylomicron, and passed into a lacteal, which is a special kind of lymphatic vessel. Any questions on how we deal with fats? Okay, so all we have left to do in chapter 34 really is make sure we've got it. Make sure we've got the big picture, right? So again, I am assuming that even if we never did this chapter together, you guys probably could have listed all these major functions for me. We eat food, break it down into smaller pieces, absorb whatever we can, and poop out the rest. Like, that's what we've been really focused on this entire chapter. Just, of course, adding more details about this, how this goes along the way. We have followed almost every step of the way how digestion occurs with like a two-pronged approach. We got to physically break the food up into smaller pieces by chewing it, the stomach churning it around, etc. But also what we really focused on for those last 3 slides was pulling together all the different enzymes that we make and use along the way to help us break big molecules into little ones that can be absorbed by those cells in our small intestine. Again, what we did in this chapter is we just followed the path of food as it moved through all of these major organs of digestion, and we stopped along the way essentially to appreciate the important contributions being made by the 3 accessory organs. The liver, which makes which makes bile that could be stored in the gallbladder. Remember, bile helps us digest fat. And the pancreas, which is secreting the sodium bicarbonate to neutralize the stomach acid, and is also making and secreting that whole mixture of enzymes, amylase, lipase, trypsin, to put as well into the duodenum, the small intestine, to allow us to finish digesting our food. Any questions on our digestive system or chapter 34? Okay. I am guessing that those last 3 slides we did about chemical digestion, I feel like that's the part that's usually newer for people. Maybe they haven't learned those enzymes before. And that's why I brought you another assignment to help you make sure you've got the details for that. Okay, so I'll pass it out probably at the end of lecture before we go to lab, so that you'll probably just take it home, I'm guessing, as homework again. Work your way through it, making sure you catch all the details. And we'll go over it together on Wednesday to make sure everyone got it okay. Questions on chapter 34 before we move on to chapter 10? Yeah, go ahead.
Actually, but kind of on all of the chapters.
That's okay.
So should we assume that we only weren't focusing on things from the slides? Like if something were to come up in the book, like a different vocab term or something that was never used?
Right, ignore it. Yep. And I would say that's been true all semester really, but especially for these chapters where I've hacked so much out of what I would normally have covered, really like focus as your starting point on what did we actually talk about together. I really don't ever, in my opinion, put things on the test that we have not been through together. I always try to focus on the most important stuff. Okay. All right, other questions? Okay, so I'll get us out of chapter 34 and we will roll right into chapter 10. Chapter 10 is once again about cell division, which we already did in chapter 9, right? So what's the deal? Why is there another chapter about this? As I mentioned in chapter 9, almost all the cells in our bodies are made as a result of the cell cycle or mitosis. But I mentioned at least a couple times that the only kinds of cells we make, not as a result of the cell cycle, are sperm cells and egg cells. Because those cells have a very different purpose and intended use than any other cell in our body, we make them in a different way. And to make sure that we make them properly, we make them in a different way with a slightly different form of cell division called meiosis. Now, what we're obviously talking about if we're talking about sperm cells and egg cells, we're getting to the idea of sexual reproduction. And I'm sure everyone at this point in their life already knew sperm cell and egg cell are going to come together during fertilization to make a baby, to make the next generation. So what we're really thinking about in chapter 10 is how does that process really happen? How is that going to work? We first have to make a sperm cell and egg cell. And just like I stressed in chapter 9, right, I stressed that mitosis or the cell cycle is a high stakes process because you're making new cells. This is also a very high stakes process, right? If there's some problem with how a sperm cell or an egg cell is made, who's going to be affected? The baby. A new member of your species, your family of course, but your species would ultimately be affected. So our body wants to make sure that we're going through this process of making sperm cells and egg cells properly, just in case they get used essentially for fertilization. Now, what we're going to do in this chapter is we're going to notice a lot of similarities. Because even though meiosis in chapter 10 is different from mitosis in some pretty major ways, a lot of the vocab, a lot of the steps, a lot of what's happening are going to look and sound very reminiscent of what we saw in chapter 9. And I always tell people this could be a good news or a bad news scenario for you. The good news is you've heard the vocab before. You have an idea about what chromosomes are, and you know, like in general, what happens during prophase and anaphase and all of that, right? Like you've seen it before. You have a sense of connection to it. But that can become bad news if you let it, right? Because there are so many similarities, you really have to make a point to get at what the differences are, to make sure you know which is which and you don't confuse yourself. We're not even going to get to the steps of meiosis today, I don't think, because we're going to do some legwork ahead of time. We're going to establish, like I always like to do, right? I want to hand you your map so you know where we're going. We're going to establish the big picture first, and there's quite a bit we're going to talk about before we look at the steps that go into this. A lot of what we have to talk about early on, genetic variation. I probably use that term like in passing in chapter 1. Genetic variation is what I always say is like, look around the room, we're all a little bit different from each other, right? And what we'll connect with here, especially kind of later in the chapter and bridging into chapter 42, that's one of the big benefits of sexual reproduction, is that just in the act of making a baby, you're making a baby that's different from any other baby that has existed before, essentially. We'll talk about why that is so beneficial as like a form of reproduction, and we'll talk about how we accomplish that as a species that reproduces sexually. Yeah.
Speaking of being unique,
yeah.
Every person in the entire world has a different iris. They're all different.
Oh, that's cool, like fingerprints? I actually did not know that.
Like our irises.
The
like
pattern?
Your iris, everyone's iris is different, like unique specifically to them.
That's cool. Even identical twins?
What?
Even identical twins? I don't.
Sure about that.
Yeah, I'm not sure about that either.
But like,
that's pretty cool. I did not know that.
My cousin's a biologist. He told me that like,
yeah.
That every single person's iris is unique to them specifically.
That's
no
cool.
One else has their iris.
That's very cool.
No one else does.
That is really cool. Yeah.
So if you want to feel
And so,
something,
yeah, you just look at your eyes. Yeah. And we're going to talk about some stuff related to that when we get into genetics in the next like chunk of the class. What we're going to learn about here is like how we get to that point of even developing fingerprints or irises or whatever. So we're really going to track like what is the dad putting into the sperm cell and what is the mom putting into the egg cell. It's just DNA, right? It's those chromosomes we thought so much about in chapter 9. They become even more important here. And that's what we're going to be tracking once we get to these steps on Wednesday. How do we divvy the chromosomes up in the right way to make sure the right number from the right parent are going to be pulled together when fertilization happens? What's going to go into it early is thinking about just how much we mix things up. You make many sperm cells or egg cells, whichever, over the course of your life, and no two of them are ever the same. Which I think, to me, like after having kids, that like really hit me in a different way. Like the kid that I had when I had them, I never could have had that kid at any other time because it would have been a different egg cell or different sperm cell, whatever. Like, I don't know, it just makes me appreciate like how unique and beautiful we all, that sounds so cheesy to say, but it never could have happened at any other time because of how sperm cells and egg cells are made so uniquely. And so that's what I want to establish right away is like, how is that the case? When all of your sperm cells and all of your egg cells are your cells with your 46 chromosomes in them, how the heck do we mix it up so much to make sperm cells and egg cells that are so different even for one person? That's what we're going to talk about in the early part of the chapter. Once we know what's going on there, then we'll talk about, okay, how does this play out? We'll look at the steps, the order they happen in, what happens in every step. And then, because like I said, there are so many similarities, there's a whole section of chapter 10 that's just a point-by-point comparison. We'll make sure we appreciate the differences. We'll make sure to highlight them, not only along the way as we go, but after we've seen both of them, we'll make sure we understand what's different between the two types of cell division that we do in our bodies. Of course, we have to be able to do both types of cell division throughout the course of our life, like from one generation to the next. If we're going to pass successfully to the next generation, you must be able to do both meiosis and mitosis, so we'll see how both fit in. This whole section of the chapter is going to go away. which is a large chunk of slides. Again, I know everyone's sad to not have as much to study for the test next week, but this is something we're going to let go of, which kills me because I find this really interesting. But to make room for what we have to cover, we're going to let some of that go, most likely all of it. Okay. So let's talk meiosis in broad terms first. I've already said the first bullet point there in chapter 9 and today for chapter 10. Meiosis is a special form of cell division that we only do in our bodies for the sole purpose of making sperm cells or egg cells. So this process only happens in the testes or the ovaries, nowhere else for any other reason. This term gamete is going to come up in multiple chapters for us as our first crack at it. Gamete just means sex cell. So I could say gamete and you would in your brain translate sperm cell, egg cell. Or I could say sperm cell or egg cell and you would just know, oh, she's talking about gametes. So really the point of meiosis is production of gametes, right? Whatever kind you make in your body, that's what meiosis is for. Now right away, big picture, what's going to be different here? Our outcome. Our outcome is going to be different because even though we're doing cell division, and maybe I'll preface this by reminding you what I kept stressing in chapter 9 for mitosis. I tried to say over and over again, right? Whatever cell you start with, good, bad, or ugly, that is exactly what you are making. Take that starting cell for mitosis, put it on a copy machine, and you're spitting out an identical copy. Not the case here. The big scale change we're going to see is that in this process, the daughter cells only wind up having half the normal number of chromosomes. And this is where we're going to make use of those terms that we learned in chapter 9, diploid and haploid. Remember what those terms refer to? The number of sets of chromosomes you have. We have 46 chromosomes in most of our cells. That's our diploid number. We only have 23 different kinds of chromosomes, but most of our cells have 2 of each. Why? The one you got from your mom and the one you got from your dad. There's like a pair of them, right? All the cells in your body, including the precursor sperm cells and precursor egg cells you have, they're all diploid. But through this process of meiosis, we're going to specifically and in a very pointed way cut those number of chromosomes in half. So that when you make a sperm cell or when you make an egg cell, we're very purposefully making it haploid. So it only gets one of every chromosome. And right away, I want us to think about why that's the case, okay? The sort of correct number of chromosomes for people is 46. That's the number we're supposed to have. So if you made an egg cell with 46, or a sperm cell, whatever, and your partner also made a gamete with 46, the sperm cell has 46 and the egg cell has 46 and they come together, how many chromosomes would the baby have? Yeah, too many. Too many. So if we make our egg cells like every other cell and sperm cells are made like every other cell, 46 is 46. It is not 46 for the baby, with 92, right? That baby could not survive. Most babies cannot survive even with 1 extra chromosome. You for sure can't survive with 46 extra chromosomes, right? If that would be the case, what the hell is the point of even making a baby? If you're making it with chromosomes too many that it can't survive, why go through the effort? So to make sure that the babies in the next generation get the correct number, we cut it back to half. When we make the egg cell, we only put in 23. When you make a sperm cell, you only put in 23 so that when they come together, 23 plus 23 gets the baby to the correct number of 46. That's why sperm cells and egg cells, through meiosis, are purposefully made to be haploid, because we're essentially thinking about what's going to happen during fertilization. How do we give the next generation the correct number of chromosomes? And to do that, we got to cut it back in the sperm cell and egg cell. This is the first time we're seeing the word zygote. Zygote is the single cell that results from successful fertilization. Sperm cell, egg cell, zygote. Okay, sperm cell 23, egg cell 23, zygote 46. Any questions on the big picture of what we're going to be tracking with this process of meiosis? Okay. So, true to form, there's a big overview picture. But before we get to it, I want to start talking about the chromosomes and this whole deal of variation that I raised the point of in the overview. So a lot of stuff on this slide, at least up top on this slide, I've said to you guys before. In most of our cells, which are of course diploid, we are diploid because we have 2 of everything. We only have 23 different kinds of chromosomes, but you have 2 of everything because your dad gave you one in the sperm cell he provided, and your mom gave you one of everything in the egg cell she provided, right? That's why our diploid number is 46. And because you got one of everything from mom and one of everything from dad, you have 2 of everything. You have chromosome pairs, right? So one from mom, one from dad, that's a pair of chromosomes. And we call those pairs homologous pairs. The prefix homo means the same. And there is a lot that is the same about those 2 chromosomes your mom and your dad gave you, right? Most of our chromosomes, by the way, are numbered. So the 23 pairs, the first 22 are numbered. Number 1, number 2, blah, blah, blah. The 23rd pair are the sex chromosomes, X's and Y's, whatever you got, okay? A lot is the same for those homologous pairs, but they're not identical. And that's what I want to talk about next. First, what is the same? Why are they called homologous pairs? Well, the chromosomes are the same length, like if you could look at them under the scope. They have the same amount of bases in them. They're the same length. The centromere, that spot where the sister chromatids are hooked together, is my weird way of trying to draw that little figure with my hands. The centromere is in the same position. Each of those chromosomes have the same genes on them, right? Like if chromosome number 12 has the gene for eye color, chromosome 12 from mom and chromosome 12 from dad both have that gene for eye color. However, maybe your mom and dad have different color eyes. So they wouldn't be giving you exactly the same gene. And this is really the first time we're hearing this distinction, which is a really important one for this chapter and multiple chapters that follow. Gene versus allele. A gene is a specific chunk of DNA with like one recipe for how to make a certain protein, right? It's one specific set of instructions for how your cell is going to do something or make something or whatever. An allele is like a different flavor or a different version of that gene. I love ice cream, so I always think about ice cream here, right? Let's say we were going to all take a field trip to the ice cream store, and I'm going to buy everyone 2 scoops of ice cream. It's all ice cream, but what flavor do you want? Maybe I want 1 vanilla and 1 chocolate. Maybe you want 2 mint chocolate chips. There are different flavors or versions of ice cream, and that's what alleles are. Now, the first way I talk about this is definitely an oversimplification, but I think it's an easy one to relate to, so I always go for it. Thinking about eye color, like I just mentioned, if your mom has blue eyes and your dad has brown eyes, why do they have different color eyes? Because that's whatever instructions their DNA has. So your mom gives you chromosome 12 or whatever, I made that number up, right? That has a gene for eye color, but maybe the allele she gave you is the blue allele. Your dad on his chromosome 12 from the sperm cell also gave you that same gene for eye color, but maybe he gave you the brown allele, right? It's all a gene that determines your eye color, but there are different versions of the information depending on what color eyes your parents have. So even though on the homologous pair, nearly everything is the same length, all the same genes, you likely have different alleles from your parents for at least some of those genes. And for that reason, the 2 members of the homologous pair are not exactly identical. You got whatever your dad gave you on this one and whatever your mom gave you on that one. And obviously, your parents are not genetically identical, newsflash, right? So of course you're getting different information from them. And that is based on what allele you're given for each gene that they provide. Any questions on the idea of homologous pairs, which we'll get a picture of in a minute, or the fact that mom and dad are giving you maybe slightly different pieces of information for some of your genes? Okay, I like this figure and the one that's going to come up a little bit later as well to help us make sense of this, okay? Up top here is a kind of picture called a karyotype. It is just a way to visualize all the different chromosomes we've got in our cells. And I'm saying chromosomes correctly here. Remember, chromatin, the crazy tangled version, is not visible under the microscope. So if you can see it, it must be chromosome, the tightly condensed kind. What this is useful for us in here is to connect with this idea that you're diploid. You have 2 of everything, right? First 22 pairs are just numbered. 23rd pair is a sex chromosome. This is 2 X chromosomes, okay? What I probably like more than what's up above, and by the way, your DNA is not fluorescently colored inside your cell. There have been fluorescent dyes used to stain these chromosomes so we can see them. I probably like this drawing better than the cool colored picture up top because this is helping us make a connection to the view of chromosomes we already established in previous chapter, right? Straight line version of a chromosome, remember, long and linear. What happens before any kind of cell division? We saw it in chapter 9 preceding mitosis. It also happens before meiosis, but the book doesn't make as big of a deal out of it. There's always an interphase where the DNA would be duplicated, right? That happens during the S stage of interphase, where remember, we just make an exact replica, an exact copy of this chromosome. And this is the structure we looked at. Let's check back on vocab, right? 1 chromosome made of 2 sister chromatids connected at their centromere, and the kinetochore protein surrounds it. Okay, all of that should be familiar. If it's not, you got to work on chapter 9 vocab some more. Okay, what we're adding in is simply the realization that, oh, we had a 12 from dad that duplicated, and our 12 from mom also would have duplicated at the same time. The book pretty consistently uses this blue for dad, red from mom coloring, okay? So what we've now got a visual on is what a homologous pair looks like. Homologous pair just means 12 from dad and 12 from mom. Remember, they're both 12s, they have the same genes, the centromere is in the same spot, but it's not identical because maybe your dad has blue eyes and your mom has brown eyes. So the information with the alleles could be a little bit different here. And that's the one new term I really want to highlight here. Non-sister chromatids. You guys know what sisters are, right? The 2 red ones are sisters because they're identical. The 2 blues, it's not labeled, but the 2 blues are also sisters because they're identical. But the blue 12 from your dad and the red 12 from your mom, they're both 12s, but they're not identical, right? Blue eye, brown eye, whatever. They're not exactly identical. So we use this term non-sister chromatid to help us track, okay, it's the same chromosome, but they're not identical because your mom and your dad gave you different copies. Now, obviously, if I'm introducing that term, non-sister chromatid, we're going to need to use it at some point. And I like that they introduce it to us here before we see it really become relevant in the next couple of minutes. So I'll come back to that point. By the way, this homologous pair, not always hanging out together. Like number 12 from dad, number 12 from mom could just be floating around in a different part of the nucleus doing their own thing. During meiosis, we're going to see them come together. And so this structure, when you've got the 2 X's together, that gets a special name. Actually, there's 2 different names for it. They're both written later, but just call it what it is from the beginning. I'm a fan of that, right? This structure is either called a bivalent, bi for 2, because you have both members of the pair together. The other word for this is tetrad, tetra for 4. 1, 2, 3, 4 chromatids of the same chromosome essentially together. And we'll see that forming in just a couple minutes. Any questions about the fact that our diploid cells have homologous pairs of chromosomes? Okay. I feel like it's gotten a lot colder in here since we started at 10. Okay, I regret taking my coat off. All right, so overview of meiosis. Here's our big picture. You guys always know in every chapter they do this, so let's see if we can track what's going on. Way up top there, I'm too short to reach it. Always look at the cell you begin with. And what I specifically want you to notice is this little box off to the side. When you see 2N like that, that means diploid. So they're drawing your attention to the fact that this precursor sperm cell or precursor egg cell, just like every other cell in your body, is a diploid cell. How can you relate to that? How they're drawing it, now we know what the red and blue mean. Notice there are 2 short chromosomes, one from mom, one from dad, and there are 2 long chromosomes, one from mom, one from dad. You guys know how to count chromosomes, as we did that in chapter 9. So if you count, there are 4 in that cell, right? 2n is 4. It's a diploid cell with a total of 4 chromosomes. The blue ones came from dad, the red ones came from mom. It's a diploid cell. And I'm specifically pointing that out because we heard already that when we make sperm cells and egg cells, we're going to purposefully make them to be haploid. So once we've gone through this deal, I'll draw your attention to what we're ending with at the bottom. Now, what makes meiosis really so different from mitosis? We're doing most the same stuff, but we're going to do it all twice. We're going to take these cells and send them through 2 rounds of division. That's why we're going to wind up with twice as many daughter cells. Right, in mitosis, we only made 2. Meiosis, we're going to make 4. And each of those 4 daughter cells, as I've already referenced, have half the normal number of chromosomes. We're making haploid gametes. Remember, 23 in all the egg cells, 23 in all the sperm cells. That's why you're seeing meiosis 1 and meiosis 2, 2 different rounds of division. Since we just learned what homologous pairs are, right? Number 1 your dad gave you, number 1 your mom gave you. Hopefully when I describe that what's going on with meiosis 1 is you are separating the homologous pairs, right? Look how they're lined up here in meiosis 1. We got 1 kind of chromosome lined up in its pairs, and the next chromosome lined up in pairs. So in our cells, really, this would be 23 pairs lined up right down the middle. And when we grab that and pull, this time those 2 chromosomes are being pulled apart. So number 1 is here, number 1 is over there. At this like halfway point, these cells are already haploid, right? This cell only has 1 short one and 1 long one, whereas when we started we had 2 short and 2 long. But you also know that the chromosomes in these cells are not like normal chromosomes yet. They still look like X's. That's what meiosis 2 is all about. We're going to rip apart the sister chromatids, okay? So we're going to rip apart those 2 long blues, rip apart those 2 long reds. Over here, rip apart the 2 blues, rip apart the 2 reds. This is how we wind up making 4 daughter cells, again, that are all haploid. When you see N or 1N, usually don't write the 1 if it's just a 1, that means haploid. And count how many chromosomes are here? Just 2, 1 short and 1 long. Again, contrast that to what we started with. We had 2 of everything. It was a diploid cell. Now we have haploid cells. The other point I really, really want to drive home early on here is that those 4 daughter cells we're making, which remember what they are, we've made gametes. Those are sex cells, either sperm cells if you have testes or egg cells if you have ovaries. Those 4 daughter cells are all different from each other. Even though they came from the same parent cell, all 4 of these are different. And here's where the overview figure does us a disservice, because those 2 right now look the same, even though they're not. These 2 look the same, even though they're not. At least where you can see a difference is by comparing these 2. This one has a short red and a long blue. This one has a short blue and a long red. Okay, so at least we can tell they're a little bit different. We're going to see, probably to start class on Wednesday, how all the daughters really are different from one another. Any questions on the idea that we're going to watch our starting diploid cell go through 2 rounds of division during meiosis so that we wind up with 4 haploid daughter cells? Okay, keep my eye on that clock. I think let's stop there actually, because it will take me longer than a minute to talk about this slide. And I want to make sure I give you guys all a copy of that chapter 34 assignment specifically before we get to lab, so that if you want to, you can keep it put away and not get rat guts all over it. That was more extreme than it needed to be, but I wanted to make the point, I guess. So I'm going to pass this out to you guys now. This is specifically looking at the last 3 slides of chapter 34, going over the details of chemical digestion. Just like it was in the slides, I broke it down into different paragraphs. one for carbohydrates, one for proteins, one for lipids or fats. And because this is the information I feel like is new for most people in chapter 34, I specifically wanted to give you guys a way to make sure you're sorting through it properly and not missing any of the details. Okay, so treat this like homework. You might have time at the end of lab to work on it a bit in your groups if you want to, but if not, take it home and bring it back on Wednesday. If you were done with the chapter 9 assignment, I'll say give it to me in lab. If you have questions about it, we can maybe go over them. I forgot I meant to do that after break. We can go over some questions about it if you have them right when lab starts after the quiz. Any questions before we head to lab? Okay, so pre-quiz on lab 11 at 12 o'clock when we start in lab. Okay, go ahead and pass them up. Before I forget, I want to talk about the chapter 9 assignment, the mitosis assignment you guys took home last week. Does anybody have any questions about that they wanted to go over? How did it go? Did you have a hard time with it? Did you figure it out? Like anything about the assignment on mitosis, because that one's due today. Yeah, go ahead and get it out if you want to. I know it might be in your bag. Feel free to get it out. We can look at it. We can go over any questions you might have, because I'm going to collect those from you guys so I can get your grades into Canvas. I'm going to try really hard to have those graded before Wednesday, so that you can take them back and look at them as you're preparing for next Monday's exam too. So just wanted to see if you had any pressing questions before you turn them in. Okay, so at any point, if you're done with it now, feel free to just pass it up. If you didn't have quite everything filled in and you wanted to talk to your normal group about it, just get it to me before you leave for the day. Sometimes day one of the RAT takes the entire time, which is why I wanted to at least mention it in the beginning so you didn't forget about it. But if you had a couple blanks that you wanted to talk to your group about, you can do that at the end of class. Just make sure I get it before you go. Okay, with that, I guess said and done, I want to move on and talk about, I'll grab them once whoever wants to pass them up has passed them up. Once I have that all tackled, thanks. What I really want to take a few / several minutes to go over with you guys to start the RAT lab is to make the point, thanks, that I initially had made in lecture, that we are spending a total of, don't just copy off someone else's. If you're going to talk about it, that's fine, but don't just blindly copy off someone's. That's not cool. Okay. Okay. Thanks. The fact that we are going to spend 3 whole days of lab on this rat dissection. We've done an entire lab in 1 day or maybe 2 days before. We've never spent 3 whole days on one lab. And like I said in lecture, I'm making a big deal about this, so you know it's a big deal. Why I make such a big deal about it is, I know you just had your first lab practical, like the last time I saw you, but you should mentally shift gears and start thinking about how you're going to prepare for the final lab practical. And the fact that we're spending so much time on this lab and there's so much in this lab, you better believe it's going to be reflected that way on the final lab practical. In the past, I've typically set up 5 or even 6 stations from the RAT lab, meaning 10 to 12 questions, meaning 20 to 24 points of your final lab practical are coming from this lab. And this is the first one that we're doing. You'll be the farthest removed from it timing-wise. So this is my upfront encouragement that you use every minute you've got to get as much as you can out of doing this dissection. And I say that specifically because I feel like sometimes people are just like, let's do what we got to do real quick and just leave really, really early. Because, hey, I've technically crossed off all the boxes. I've checked everything off the list. Don't do that. Like, even if you think you've seen everything, go back through it again like another time or 2 or 3, whatever you have time for. Because what you put into it now is the payoff you're going to get at the end, really. So again, what I want to do before we even get into it is talk to you about what's in the room, what's in the book, kind of how we're going to approach this. Okay, so I'm going to open my book up to the beginning of the RAT lab, which I have to find, on page 91. Okay, so quick little intro here, basically just giving you the overview of what we're going to be looking at. And then right away on page 92, we start in with part A. Part A is essentially what we're going to tackle today. External anatomy, looking at parts on the outside, and skeleton, I'm going to come back to the book in a few minutes to really explain what we're doing and how to tackle it, because we're also going to start part B today as well. I'll come back to that so that what you actually have to do is fresh in your mind when we get started. First, I want to show you what's here. Before I forget, my friend over there, skeleton model. Part of this lab is going to be to know pretty much all of the bones on that skeleton. If you've learned bones before, that's no big deal. If you haven't, there are a lot of bones, and that's just one part of this. There's a figure I'll show you later, which lists all the ones you have to know, and I'll encourage you, hey, at some point, whether it's today, Wednesday, or next Monday, whenever, bring that figure up here and touch every bone. Make sure you know every single one, because you could be asked about any single one of them, okay? Over here, we've got a bucket, which I grotesquely refer to as the parts bucket. Any part that you cut off your rat completely will go in this bucket. Today, what that's going to be, and I'm just going to be real honest with you guys, I think today's the worst day. So if you can get through today, smooth sailing from here. Why I think today's the worst is we have to get the skin and the fur off of the rats so we can see the muscles underneath. So you're going to be skinning your rats today. Once you get the skin off, it goes in the parts bucket. Okay, today's the worst day. I'll say more about that in a minute. Okay, this over here, liquid waste container. When you get your rat, all of them are in sealed plastic bags with a small amount of preservative fluid. So you'll cut your bag open, you'll get your rat out, and you'll come, bless you, dump the liquid into the liquid waste container. That's the only time we'll be using this all week. This is, I think, where the worst smell is coming from. So like after today, I'll make sure this is capped, and that'll cut back on the stench a little bit, hopefully. Over here, I mentioned bones are important. Little rat skeleton here mounted in this container, and figure that goes along with it. You're not going to dissect your rat down to the bones. So if you wanted to see what they look like on the rat, this is how you could do that. Your focus, which I will stress, which your lab book says, is to learn them on a human skeleton. So this is cool just to see, but you don't have to know anything off of this. Again, now that you've been through a lab practical, how in the world would I ask you what any one of those bones are? Because it's totally enclosed in a case. I can't ask you that. It'd be much easier for me to ask you on the human skeleton. We also have over here preserved rat in a jar. You can see the bones on the cartilage, another view if you want to. You guys know what a big deal I usually make about models. I don't make a big deal about this one. Again, put yourself in my shoes. At the final lab practical, would you rather show a student a plastic model or an actual rat? Actual rat. So you're going to see multiple rats on the final lab practical. I'm holding my hand here because the organs are removable, so if you want to come see like kind of how they fit together, that's a good way to do it, I guess. And there's a diagram that's labeled here. I think a better way to look at it is in the actual rat you're going to dissect, so it's fine, but it's not the most useful in my opinion. Back here we have gloves. We're supposed to tell you guys that students are limited to 1 pair of gloves per lab period. So like if you think you need to use the restroom or whatever, do that before you put your gloves on. You can take them off carefully and not contaminate yourself essentially, but you're supposed to only use 1. I don't usually see people, you know, using like 5 pairs per lab, so it's fine. If you realize you have to go to the bathroom, take them off, get a new pair. Just I'll look the other way, but don't go through a whole bunch of them. Also back here we have dissecting trays or pans, which are at the present moment clean and dry and lined with 1 paper towel that's been unfolded into the bottom of it. This is how you will leave it when you are done with it. Clean, dry, lined with a paper towel. Next to that we have dissecting kits. This has all the tools you're going to be using. Each group, you're going to work in groups of 2 or 3, no more than 3. Each group will get a rat in a pan. I'll pull the bags out. They're in the bucket right now. I'll pull them out and lay them in the trays. So you can just come back, get your gloves, get a tray, get a dissecting kit. Over here there are also some pins, T-pins, because they look like the letter T. You don't need any of those today. On Wednesday and Monday we'll use them. Alcohol, prep pads. This is kind of a fast and easy way to clean your tools when you are done. Just like the trays are clean and dry, so must your tools be when you put them back. Inside that kit, by the way, you're going to find a scalpel, which is clearly sharp, so be very careful. not to cut yourself. I actually don't use the scalpel, not even once, the whole time I do this dissection. And I'll show you how that's the case later on. There are tools I prefer instead that are, I think, a little safer to use. I've had one student get cut during this lab. So just as a precursor, here's what happened. Her lab partner was holding the rat while her lab partner was cutting. That is like the worst idea ever. If you're holding and cutting, if you slip, you feel it right away and you have the instinct to move your hands. If someone else is cutting and they slip, you don't know till they cut you to like pull your hand away. That's what happened. Her lab partner cut her. Accidentally, of course. It wasn't it was just a little nick right through the glove though. So don't ever, like if you're the one cutting, whatever tool you're using, scissors or scalpel, no one else has their hands on that rat. If you do get cut, of course, let me know right away. I'll help you get a Band-Aid. We'll call campus police and file an incident report or whatever. Just be careful, right? That scalpel should be, if the previous class listened to what they're supposed to do, it should be blade side down, because you want to make sure that when the next person opens the kit, they're not going to cut themselves right away on it. So when you're done, you can either wash your tools in these pans of soapy water and then rinse them and dry them, or you can wipe them off with an alcohol prep pad, which might be quicker, I think, honestly. And then as you put them back, you might want to actually take a picture of how the tools are in here before you get to work, because that way you'll know where to put everything back when you're done. It'll fit in there a lot easier. And then make sure you put them back clean and dry. There's a Sharpie back here and some baggies. Guess what's going in the baggies? Your rats. You're gonna put the rat in there when you're done with it today. You're gonna give it super funny name, or just write your names on it if you wanna be boring. Put your rat in the bag. You're gonna give it a squirt, not of bench cleaner. Don't spray it with Mr. Clean or whatever that is. This is called humectant spray. It just keeps the rats, I don't want to say fresh because they're dead rats, but it keeps them in good working condition over the 3 days that we're going to use them. Okay, so put it in the bag, 1 or 2 squirts, seal the bag, and you'll put it right back in the bucket. Okay. Questions about what I've shown you around the perimeter before I head back up front? Okay, so there are a couple more things on the front bench that are going to be useful as we complete this lab. One of them that I don't think will be that useful, but I want to mention, in this box, which I'm not even going to open, there is an already dissected rat that's been dipped in plastic to preserve it. It's another view, like better than the model, not quite as good as your actual rat, but if you want to see it, it's here. I don't think you're ever going to see that from me again. Read between the lines. What will be more useful to you during this next week and a half in lab are these pictures that are up front. These are color pictures, obviously, that are labeled of already dissected rats. There are numbered parts here. The key is in the middle. Why do I think this will be useful to you? Not today, but Wednesday and next Monday. As you guys know, there's one of me and there are 25 of you. So there's going to come a time when your group has a question, what is this? How do I find whatever? And you might be like the second or third group in line waiting for help. So this is a good kind of bridge the gap. You can look for it on here and try to piece it together with what your rat looks like. These are available for sale in the bookstore if you wanted your own. I probably shouldn't say this for copyright reasons, but if you took a picture of this and I didn't see you, I couldn't tell you not to. So that's an option as well. The other thing up here that I do want to mention specifically is this stack of index cards, laminated index cards. I'm touching this with no gloves. I would not recommend you do that. I'm going to wash my hands in a minute because I assume there are rat guts all over this. What is this? This is a list of major parts you're supposed to get out of doing this lab. Bones, muscles, organs, blood vessels. That's a lot of parts. I actually had these made for both the lab rooms that have 1,000 in them, specifically several years ago, because this is how you can check yourself, right? If you feel like, okay, my group has been through everything, I think I got everything, grab some cards and quiz yourself. See if you can find every one of these parts, if it's a bone on the skeleton, if it's anything else on your actual rat. Hopefully you can. If not, you still have some work to do. Really why you're going to care about these 2 magic words, extra credit. Next Monday, on our last day of the rat, it's just going to be basically a review day. You'll look at everything you've seen again. And on that day, whoever wants the chance, which I hope would be everyone because it's extra credit, you got nothing to lose, I'll blindly let you pick 5 cards. So face down, you don't know what you're grabbing. For every one that you can identify correctly, again, bone, you take me over here. Anything else on your rat, you get a point of extra credit. So up to 5 points. These will not be the only extra credit points offered. There are more coming at the end of this semester. I usually hold most of it till the end because that's when people care the most about it, quite honestly, and when you see the biggest effect on your grade. We are limited in how much extra credit we are allowed to offer. Like as a department, we've agreed on a certain point, and I max out what I'm allowed to offer because I know my tests are hard, so I want to give you some ways to make up for it. Okay, so I usually offer about 40 points total. This is just 5, so there's more coming later, just as a heads up, okay? Any questions about what I've said so far? All right, so now that you know kind of what's in here, let's talk about what today is going to be. I already said you're going to work in your groups of 2 to 3 people. I think I said this like the first day of class, but that's been a while, so I just want to repeat. I get no joy out of forcing people to touch a rat if they don't want to touch a rat. In other words, if you are not about this, if you are grossed out, if you don't want to be, if you're like, oh, this is disgusting, don't touch the rat. I'm okay with it. As long as you know that you're still going to be held responsible for knowing all the same stuff as everybody else, right? Even if you don't physically touch the rat, you got to learn all the parts of it and what's going on. So why I always like make a big point of this at the beginning is if you're in a group of 2 people and you are not touching the rat, you want to make sure your lab partner is okay with the one like doing the heavy lifting, right? If I don't want to touch it and you're not going to touch it, we should not be in the same group as only the 2 people in the group, because then who's gonna touch it? Someone has to touch it. So just as you're getting into your groups today of 2 or 3 people, just say like, yeah, I don't want to touch it, or hey, I'm okay with it. Like make sure you're in a group of 2 or 3 people where someone's okay with whatever that is going to turn out to be. Okay, questions about that? Also, every group of 2 or 3 people, you can't do more than 3 because 4 is too many people to get around like a dissecting tray. You won't be able to see everything going on. Your rat is either going to be a male rat or a female rat. Okay, the bags will be labeled so you know what you're getting, but you're going to get one or the other. And your job is to make sure you see both, which means at some point you're going to have to look at a rat that's not yours, that's not your group's. I think the easiest way to handle this is think about your normal group of 4 or 5 people. You're going to split into probably a group of 2 and a group of 3. One of you get a female, the other one get a male, and then you can just switch rats with each other or help explain it to each other. That way you're not like, who has a male I can look at? Who's got a female? Right next to you, you'll know there is a rat of the opposite sex. Let's start looking at the lab book to see what we're going to be doing today. I'm starting us off on part A, which I mentioned already is on page 92, external anatomy and skeleton. So if you kind of look real quickly at what is on page 92, it's basically talking to you about like external parts on your rat, right? That's what external anatomy means. It's going to have you look at the teeth. There are 4 of them. They're all incisors, 2 up top, 2 on the bottom. You'll see them when you get your rat. And then what I really want to talk about is number 4. It's the point I just made about, hey, you're going to have either a male or a female. This is going to sound really strange to say, but I don't care, whatever. It might not be as obvious as you think it is to tell which is which. Everyone always thinks, just look for the penis. Like, obviously, the males will have one and the females will not. The penis is actually not the easiest way to tell. It's kind of small on the rats. So what you want to look for instead is the scrotum. The scrotum or the sac is much larger than the penis, and the males have one and the females do not. That is the quickest, easiest way to tell what's what. Again, the bags will have a label on them, so you're going to know what you're getting. Before you do anything else, after you get it out of the bag, get the male and the female right next to each other and make sure you guys can tell them apart. This all sounds rather funny and sort of obvious, like who can't tell them apart? I know people can't, because on a lab practical in previous years, I have asked the question. I just had a rat laying there. What is the sex of this rat? Not everyone got it right. And it's like a 50-50 shot, right? And not everyone got it right. That's how I know it's not as obvious as people think it will be. So again, I'm making a big deal about it so you can make sure you know how to tell them apart. On page 93 in number 7, again, it specifies bones on a human skeleton, which is up over here, the model. There's a whole list of bones here on page 93. I want you to flip to page 98, which is the figure that goes along with the list of bones on 93. This is the figure I mentioned earlier that at some point, whether it's today, Wednesday, or next Monday, at some point, you and your group, you and your partner, whatever, bring this up here and like touch every bone as you look at it. There is a greater connection you can make by touching it versus just looking at the figure. Again, pretend you're me. If you were designing the final lab practical and you wanted to ask students about a bone, what material would you put in the room at a station? Ding, 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 right? If I wanted to ask you a certain bone, can you envision, now that you've been through a lab practical, how might I ask you about a certain bone? With an arrow taped to it. Again, you can write the test yourself, especially once you've been through one. This is why I want to say, like, first day into the second half or whatever, do this every lab. How could she ask me this question? What could she ask me? What material could be at a station? You can write the lab practical yourself, okay? Obviously, the challenge is there's a bunch of bones. How do you know which one I'm going to ask you? You don't. That's why you have to know them all, okay? There are not many places where I can trim stuff down because there's a lot in this lab. Wherever I can, I will try to cut out little stuff. On the bone page, I'm going to cut out a couple things for you. So on page 98, in that offset in the upper right corner where the cranium is, the only two you're going to keep that you should know are the two jaw bones, maxilla, which is the top jaw, and mandible, which is the lower jaw. All those other parts of the skull, you can cross off, meaning you don't have to know it, meaning I would never ask you about it. Cross off frontal, cross off parietal, cross off temporal, cross off occipital, cross off zygomatic. Again, the only parts on the head you have to know bone-wise are maxilla, which is the top jaw bone, and mandible, which is the bottom jaw. If you get them mixed up, a lot of people do, how I remember, maxilla, max, like maximum or best or most, maximum is top, okay? Maxilla, top, mandible, bottom. Once we're in the bigger skeleton picture, see off to the left how it says vertebral column? That just means everything by the vertebrae. And if you look for the vertebrae labels, there's one cervical vertebrae, which is in the neck, thoracic, you guys know that means chest cavity, right? Thoracic vertebrae are in the chest, and then lumbar vertebrae are in the lower back. All you have to know about them is that they're vertebrae. I'm not going to ask you what's cervical versus what's lumbar. So you could cross off just the word cervical, leaving vertebrae. You could cross off just the word thoracic, leaving vertebrae. You could cross off just the word lumbar, leaving vertebrae. I think that is one of the easiest bones then because it's like all the vertebrae in your whole spinal column. All the other bones are good. They're all staying. You should know them all, okay? So if you already know them from some other previous experience, awesome. If not, that's a lot already just with the first thing, okay? Go ahead.
Are you going to give us like a worksheet, like a fill-in-the-blank worksheet? Or?
I usually don't. I can look for some.
Oh, yeah.
If you think that would be helpful, I can look for some. I also have had students tell me before about apps they've found. I'll try to dig through my email. I probably saved it. Like an app or a picture you can go through and kind of quiz yourself and label it, which they found useful. So I will dig that up and try to post that. Speaking of posting, if you haven't seen it in the lab module, just like there was a Lab Practical 1 review sheet, there's also one for 2 that you could print off this week and shove in your book and check it every lab we do. And there's also a separate page titled Rat Dissection Resources. I posted links there to multiple websites where you can see diagrams. Some you can toggle the labels on and off so you can quiz yourself. That page also has all of these terms listed. So if you wanted to like try to study for the extra credit ahead of time, you could look at the terms that are on the cards and make sure you know where those parts are. Other questions before I talk about Part B a little bit? All right, Part B on page 94. This is essentially everything else, like beyond the bones and the external. This is where everything else is contained. Part B1 is the only thing we're doing today, muscles. Okay, now, what 1A B1A, whatever, dissection. That's talking you through how you're gonna skin the rat. I'm gonna do a demo for you guys. The goal of today is not to like be the best rat skinner that ever existed. We just wanna get the skin off so we can see what's underneath. I have skinned way more rats than I ever would have believed possible in my life. So I am fine helping you guys do this part. I don't care if I skin all 10 rats myself. I'll help you as much as possible to get you through it. We just got to get the skin off so you can start looking for the muscles underneath. So I'll do a demo so you guys know how to get started, and then I'll just essentially run around from group to group helping whoever wants help, okay? If you want to do it yourself, awesome. If you get stuck or have a question or whatever, I'll help you do whatever part of it you want help with. Once you get the skin off, then your job is to look at muscles. Notice under muscular system B, anatomy of a muscle, it says optional. We're going to opt to cross that whole paragraph out, scribble it out, okay? What that contains is a cool description of how muscles work, but what it does not contain, which is what you actually need, is a list of the muscles you're supposed to find. Like they're not listed anywhere, which is why we're going to go right to the picture pages. Page 99 and 100 are the picture pages, okay? Why are there 2 pages? Let's look at the captions. Page 99, white rat muscle system, dorsal view. What does dorsal mean? Back, right on. So this is your rat is laying on its belly with its back up to you. These are the muscles you're going to find on that view. Notice that dotted line right down the middle of the page? The labels are at the very top. On the left, superficial muscles. These are the muscles that are closer to the surface that you can see once the skin has been removed. While on the right-hand side, those are deeper muscles that are typically covered up by some other kind of structure. They might be beneath the superficial muscles. They might be in between some of the bones. You got to usually do a little bit more digging to actually find them. Okay, so the superficial muscles should be easier to find. The deeper muscles, you should know where they are, but you might have to dig a little bit to actually see them. If you flip to page 100, same deal, but look at the label this time. White rat muscle system, ventral view. Ventral is front. So now your rat, you've turned it over. It's laying on its back with its belly up to you. These are the superficial on the left and the deep on the right muscles that you're going to be finding. Second one down on the left, sternomastoideus, cross it off. There's so much going on in the neck area that one is just really tough to find for most people. I will never ask you that. You don't have to know it. All the rest of these are pretty good, okay? Overall, what I want to say about the muscles, these figures look really nice, meaning there's clear definition between different muscles. You can tell which way the muscle fibers are running. I just want you to be mentally prepared for the fact that your rat's not going to look anything like this. In reality, the muscles on your rat are going to be some version of like beigey, gray, pink, somewhere in there. You're not going to see great definition between them. You're not going to see necessarily clear striations that tell you what's going on. They're a lot harder than these pages make them look like. I think they're some of the hardest things on the rat, until you know. What I would encourage you to do is sort of memorize what's supposed to be where, right? What do I mean? For example, let's look at the legs. On the hind legs, on the bottom legs, if you're on page 99, there are only 2 muscles on the dorsal view in the leg you should be finding. One of them is above the knee and one is below the knee. That's what I would encourage you to pay attention to. What area of the body, what side am I looking at? This is what's going to help you. Above the knee, biceps femoris. I always talk about that one because when people hear biceps, they think biceps, like in the upper arm. There are biceps in the upper arm, but if you look for the label, it's on page 100. Those are biceps brachii. Brachii, think arm. Biceps femoris, think femur, which is the biggest bone in your leg. Femur means leg. So biceps femoris is not the biceps you would usually think of in your arm. People get thrown off by that, so I always talk a bit about it. Below the knee, gastrocnemius, which is your calf muscle, essentially. If you flip it to page 100 and look at the leg, we flip the rat over now. It's belly up to us, and the only muscle on the leg, on the belly side that you are looking for is the gracilis. That's what I mean when I say pay attention to like what's supposed to be where. What's on the arm, what's on the leg, what's on the front, what's on the back. That's, I think, what will help you more than this. And even more than that, if we're thinking ahead to lab practical. I already said there'd be real rats there. Just to tell you how it's going to be, there'll be rats in dissecting trays, and I'll have a pin in it somewhere. And I'll say, what is this pin pointing to? And like before, it's all multiple choice. So it's not like I'm going to be breathing down your neck saying, find the gracilis. Like you'll have a muscle, you'll have a list of choices, right? So as long as you can do a process of elimination, you'll have a pretty good chance of getting it right, okay? People typically, like in past semesters, people typically do pretty well in the rat lab because we spend so much time because of, I think, the way the questions are set up, okay? So overall, your job today, figure out who you're working with. I'm gonna put on gloves and start pulling rats out into trays. One of our female rats is pregnant. Who wants the pregnant female? Okay, first dibs, you got her. I think she's the coolest rat. On Wednesday together, we will count how many babies she's got, because rats have litters just like dogs and cats do. We'll do a little C-section, check out what's going on with the baby, talk about the placenta and the uterus, all that. So that's, I think, a good rat to have. Record number of rats from my sections, I think was 16 in one rat. So we will see, they were really little. Once they get bigger, there usually aren't so many. We'll see what we got going on for our section. So what you can do once you figure out who you're working with is everybody come get a pair of gloves. I'm gonna pull the rats in their bags out and put them in the trays. Your first job is gonna be to cut the bag open with the scissors in your dissecting kit. Take the rat out, put it in the tray, go drain the fluid in the liquid waste container, and then throw the bag away. Then you've got your rat out with your tray, with your kit. You're gonna do all external anatomy. First group that thinks they're ready to start cutting, call me over. I'll have everyone come look at a demo of how to start skinning. Okay, sound good? Good-ish, as good as it can for a rat dissection. All right, I'm gonna start pulling these out. The bags, right above where there's a barcode, the bags say male or female, so you'll know what you're getting. Remember to coordinate with the group next to you. Looks like all the females are coming out first, so I'll start stacking them over there. Laurel, here's your pregnant female. Okay, I'll put it by the sink over here so you'll know where she is. Another female. Yeah, they're all females so far.
These are extra large. These are
Yeah, we don't have any small gloves, just medium, large, extra large. We got a male. Heard you just say it. Were you waiting for one too? I'm sorry. There should be another.
How many were you taking these?
None for today.
Okay.
This should be our last female right here, so the rest should be males. All right.
Thank you.
Oh, yep. Yeah, so we only are allowed 10 rats per lab, so some of you will have to have groups of 3 if you weren't planning on it, just so you know.
How do you know what's a boy or girl rat?
Right above the barcode, it should say male or female.
Got the boy one.
Yeah, boy? Okay. We got 2 more females and a male still up for grabs. Male, female. All right, so remember, first thing you're doing is cutting the bag open, getting the rat into the pan, draining the liquid waste into the waste container.
Good luck with today's lesson.